Hello, my name is Lim Kin Jian. The topic of the research article chosen is Horizontal Gene Transfer Contributes to Virulence and Antibiotic Resistance of Vibrio harveyi 345 based on complete genome sequencing. Vibriosis has become one of the most serious diseases to endanger sustainable aquaculture development. Wide use of antibiotics, increased environmental pollution and global climate change lead to enhancement of antibiotic resistance of Vibrio species. Virulence factors and antibiotic resistant genes can be vertically transferred and spread via horizontal gene transfer through mobile genetic elements such as plasmid, bacteriophage, transposon, integrative and conjugative elements, genetic islands. Vibrio harveyi 345 is a gram-negative gamma proteobacteria. It is multi-drug resistant and is the pathogen of vibriosis for marine vertebrates and invertebrates. It causes kidney enlargement and softening, spleen enlargement, and anal bleeding of E. lanceolatus. DNA extraction. Vibrio harveyi 345 was isolated from a disease E. lanceolatus in Shenzhen, southern China. Genomic DNA was extracted using CTAP method. Whole genome sequencing and assembly. PacBio RS2 20 kilobase template library was constructed from 10 microgram genomic DNA. Libraries were sequenced on Illumina HiSeq 4000 and PacBio RS2 sequencing platforms. Genomes were assembled and contigs were error corrected with a variety of software. Gaps between contigs were filled by PCR. General annotation. Open reading frames were predicted by Gleamer. Genome annotation was completed. Virulence factor and antibiotic resistance gene were identified by BLAST against virulence factor database and antibiotic resistant gene database respectively. Genetic islands, prophage, and CRISPR-Cas system were identified. Comparative genome analysis was conducted among 31 Vibrio harveyi strains with Vibrio harveyi VH2 as the reference strain. Pan-core genes were analyzed using BLAST method. Gene families were constructed using genes from reference strain and target bacterium. Strain-specific genes extracted were compared to non-redundant protein database using BLAST-P2. A total of 1,189 megabase clean data and 98,602 subreads were generated, assembling into two contigs with average genome coverage of 41.71 times. Genome of 6 billion base pair was generated after error correction, GATK analysis and gap filling. Complete genome of Vibrio harveyi 345 contained 2 circular chromosomes and 2 circular megaplasmids. 5,678 genes were predicted, accounting for 85.29% of the genome. 487 putative virulence factors were predicted, involving adherence, motility, regulation, secretion system, and ion uptake. 25 antibiotic resistance genes were identified. 7 gene families were strain-specific to Vibrio harveyi 345 with more than 50% identity to other species. 217 genes were specific to Vibrio harveyi 345, suggesting that a large number of horizontal gene transfer events happen in this strain. 3 strain-specific genes encode proteins which contribute to antibiotic resistance and virulence of this strain. The proteins share no similarity with any proteins of Vibrio species, but show high identity with proteins in other species. 5 antibiotic resistance genes and 1 virulence gene were identified on plasmid P345185, which show no homology with other Vibrio harveyi plasmid. This gene could be horizontally transferred, promoting antibiotic resistance and virulence. 71 genomic islands, 2 incomplete prophage sequence, 3 type 2 secretion proteins, and 13 type 6 secretion system proteins were detected that could be horizontal gene transfer delivery tools. Complete genome and information of horizontal gene transfer event of Vibrio harveyi 345 are helpful for identifying pathogenesis and drug resistance. Complete genome sequencing and comparative genomics show that virulence factor and antibiotic resistant genes can be transferred via horizontal gene transfer through mobile genetic elements. Studies on horizontal gene transfer events could improve understanding on the effects of wide use of antibiotics, environmental pollution, and climate change on antibiotic resistance. This information is useful for controlling the disease and reducing economic losses. Thank you.